Hi, this is chapter 27 of the urinary system by Frederick H. Martini. Our goals for this chapter is to identify the components of the urinary system, the structures, and we're going to focus on the function of the nephron, which is where most of the physiology that leads to the formation of urine occurs. So the urinary system includes kidneys, ureters, urinary bladder, and the urethra. Once again, kidneys is the main site of physiology. The ureters are in charge of transporting these components from the kidney, the urine from the kidney to the bladder. Um, the kidneys, in terms of anatomy, they are retroperitoneal. They are behind or posterior from the peritoneal cavity. This peritoneal cavity, which houses our digestive components. I want you to see here the three layers of the kidney that are served for um, protection and as well as attachment. One of them is going to be the capsule, that is a layer of dense irregular connective tissue that immediately surrounds the kidney. You have this adipose pad layer here, this adipose capsule, and you have a renal fascia that is contiguous with the um, peritoneal layer here, and the deep fascias of the muscles in the posterior area. From a posterior view, you can see that the left kidney is higher. It's higher than the right kidney. The kidneys are going to be vascularized by the renal artery, which leaves the descending aorta at the level of the superior mesenteric artery. And you observe here the suprarenal glands present. One of the things that I want you to keep in mind as we look at the function of the kidney is that you will have these vascular components leaving. So you have these arteries going into the kidney, and you have these renal veins leaving the kidney. In between that process, components from at the capillary levels are going to be filtered into this renal tube system that eventually is going to lead to the exterior environment. So you're taking components, filtrating components from the blood, leading into the renal tube system, and from the renal tube system, you're going to reabsorb a lot of those components, and eventually, whatever is not reabsorbed, it's going to leave through the urine. Here you have a view of the sectional anatomy of the kidneys. One of the things that I want you to focus here are here we have the cortex, in which you will find your renal corpuscles, which are going to be a part of the nephron, the functional part where the filtrate is being formed. And here you have the medulla. The medulla, you will find these lobes. Within these lobes, you will find the renal pyramids. The renal pyramids eventually lead into into the apex of the pyramid, which is known as the renal papilla. The renal papilla um, it has the look of a sifter, and all of those holes will be connecting these ducts into the calyces here. The calyces, you have your minor calyces, major calyces, and they lead to the renal pelvis, and from the renal pelvis to the ureter. I want us to take some time to look at the vascularization of the kidney, which is something that is extremely important for its function. So you will have once again, at the level of the renal corpuscle, at this very microscopic level, you will have blood being filtered into the renal duct system that will eventually lead. This is the first step to the formation of what is known as urine. So let's look at these arteries. You have the renal artery entering the kidney at the hilus. It divides into segmental arteries. These segmental arteries divide into lobar arteries. Each lobar artery supplies a lobe. Eventually, these lobar arteries bifurcate into interlobar arteries. Interlobar arteries go between the lobes. These interlobar arteries transition into arcuate arteries, which create this arch present here. And I'm going to transition to this view. Create this arch present here. And from this arch, you have the branching of interlobular arteries, and each interlobular artery will have several afferent arteries growing from it. And these afferent arterioles, these afferent arterioles are going to create this capillary bed known as a glomeruli, which resides within the renal corpuscle, which is responsible for the formation of for filtration, which gets filtered, and it's a finished step of urine formation. This is the same view. You have urinary arteries creating at the level of the nephron. They create this capillary bed known as a glomeruli, and from the glomeruli, you will have an efferent arteriole going into the glomeruli. This capillary bed eventually leaves the glomeruli or the renal corpuscle, and it becomes a peritubular. It basically the circulation circles back into the nephron in the form of peritubular capillaries for the purposes of reabsorption. Everything that we have filtered was filtered almost indiscriminately, but we have a very selective procedure in which we reabsorb the components of this filtrate. Eventually, these components go back into venules, interlobar arteries, arcuate, and eventually into the renal vein. Here we have a view of the nephron. Let's start where the afferent arteriole enters the renal corpuscle. The afferent arteriole creates this capillary bed, which is known as a glomeruli. This capillary bed, eventually there's the pressure under which this capillaries under will determine the amount of filtration that occurs. This filtration will occur in this capsular space of the glomeruli. This will lead to the proximal convoluted tubules, the nephron loop, the distal convoluted tubules, eventually to the collecting ducts, this portion of the conducting system. The collecting ducts will lead to the papillary duct and eventually to the minor calyx. And from the minor to the major to the renal pelvis to the ureter, bladder, urethra, and the exterior environment. Here are some of the functions of these different components. The proximal convoluted tubule is involved in reabsorption of water, ions, and all organic nutrients. So here you have your glucose being reabsorbed. Then it goes through the nephron loop, the renal loop, or loop of Henle. Here we have further reabsorption of water and both sodium and chloride. You eventually will lead to the distal convoluted tubule. You have once again secretion of ions. You have at this level you have secretion of ions, acids for regulation of pH, drugs and toxins. You have variable reabsorption of water. This would be under hormonal control. And at the descending, at the collecting duct level, you will have once again variable absorption of water under hormonal control, and you have secretion of sodium, potassium, hydrogen, bicarbonate, and eventually at the papillary duct, you have the delivery into the minor calyx. So let's look at the function and the regulation of the nephron. The function of the nephron is going to be the production of filtrate, reabsorption of organic nutrients, reabsorption of water, and secretion of waste products. So basically, we can summarize this as filtrate production, so we indiscriminately filtrate components, then we reabsorb the ones that we need, including organic nutrients, water, and ions. and then we secrete the components that from the blood into the filtrate that need to be that haven't been that haven't yet entered the filtrate. So this would include creatine, for example. Regulation, you have regulation at the hormonal level, you have regulation at the auto regulation as well. Um, so some of the some of the examples of hormone regulation, you have antidiuretic hormones, you have the renin-angiotensin system, you have aldosterone having an effect on the regulation of filtration within the kidney. We're looking at two types of nephrons. One of them is the cortical, another one is a juxtamedullary. The cortical is this particular nephron. It's mainly found in the cortex, and the juxtamedullary is a nephron whose renal loop extends all the way into the medulla. This allows the formation of a concentration gradient within the medulla that is that serves for the purpose of water reabsorption. So we create this osmotic gradient with sodium ions and the urea that allow water will follow will follow sodium ions. So if we put a lot of sodium in this exterior environment, a lot of sodium is going to leave the water will follow the sodium, and eventually that water will go into our circulation, which we don't see here. So this is the blood that efferent arteriole that leaves the glomerular, the renal capsule, eventually circles back all over the nephron in order for reabsorption to occur. So here you have at a cortical nephron, you have these what are known as peritubular capillaries surrounding it in an effort to reabsorb components. And at the juxtaglomerular nephrons, you have this specialized capillary bed known as the vasa recta, which is there for the purpose of reabsorption of water and other components. Let's look at the workforce of the nephron filtration. This is the glomerula, the renal corpuscle. Specifically, we're going to focus on the glomerula. This capillary bed composed of the afferent arteriole and eventually leads through the efferent arteriole. As you can see here at the level of the efferent arteriole, we will have regulation of the amount of pressure at which blood is going to reach this arteriole, because this endothelial epithelium is fenestrated. It has it's porous. So if there's high pressure, then liquid and ions and other components are going to just leave the blood and enter the capsular space here. If there is low, and we regulate the amount of pressure at this level, where there can be constriction of the small muscle surrounding this afferent capillary, so that to prevent high pressure going into here. One thing that I would like to mention is that this usually the pressure found at the glomeruli is 50 millimeters of mercury compared to the regular pressure of capillaries beds at 35 millimeters of mercury. A part of that pressure is created through the efferent arteriole. The efferent arteriole has a smaller lumen than the afferent arteriole, effectively creating a bottleneck as water gets, as blood enters this, and you have a bottleneck, a lumen here. The volume is going to pile up, and it's going to create a higher pressure. Here we have a specialization called the juxtamedullary apparatus. The juxtaglomerular apparatus. The juxtaglomerular apparatus is going to be composed of specialized cells from the distal convoluted tubule called the macula densa, and these specialized smooth muscle cells that are present in this area that are able to control the lumen of the efferent arteriole, as well as release renin and erythropoietin in order to regulate filtration. I want you to focus on, so this is the capsular space. Eventually, this is where the filtrate is being created. So this is where water is going to, if under enough pressure, fluids are going to leave this capillary, aided by the podocytes. As you can see here, we have two layers in the renal corpuscle. One of them is going to be this parietal layer, this parietal epithelium, which is contiguous with this visceral layer, this visceral epithelium. The visceral epithelium is composed of podocytes. These podocytes contain these specializations called pedicles. And here you have the podocytes and their pedicles here. They create these filtration slits that aid in the filtration of components from inside the blood into the capsular space. We also have here the presence of the presence of mesangial cells present here, which contain actin filaments in them. These actin filaments allow them to contract and regulate the lumen of these capillaries, basically regulating pressure within them. The renal corpuscle generates about 115 liters of, it filtrates 150 liters daily. The proximal convoluted tubules reabsorbs water, organic substrates, ions, glucose, amino acids, and it secretes hydrogen ions, ammonium ions, creatine, and drugs. So basically, here there's regulation of pH, regulation of weight and waste products. They can be secreted at this level, and most of the reabsorption, 60% of the water occurs here, as well as most of the absorption of nutrients such as glucose. The nephron loop reabsorbs water, reabsorbs about 25% of the water, as well as a large amount of sodium and chloride. And it creates a concentration gradient within the medulla, which we will see how it aids in the reabsorption of water. The distal convoluted tubule reabsorbs water. It's under the stimuli of ADH. It also reabsorbs sodium under aldosterone stimuli. Basically, by reabsorbing sodium, water will follow. So it will reabsorb sodium and water, it's a method of regulating through antidiuretic hormone, regulating how much water is lost. The collecting ducts, once again, under hormonal stimuli, they are able to reabsorb water, and this is kind of like the last chance to reabsorb, and they secrete potassium and hydrogen. So this urine production maintains homeostasis, regulates blood volume composition, and it excretes waste products such as urea, creatine, and uric acid. We mentioned about these basic processes: filtration, reabsorption, and secretion. I want you to keep in mind where is the initial point and the end point of these processes. The initial point of filtration initiates in the blood and ends up in this tube system, which it starts at the capsular space. Reabsorption that initiates in this tube system within the kidneys and ends up being reabsorbed into our blood. Secretion, reabsorption, the filtration is kind of an indiscriminate, almost driven by pressure, but system of exchange. But reabsorption and secretion is highly regulated. This is done mainly through transport channels. And so whenever it comes to secretion, we are choosing what we lose. We're like, do I want to lose this and we have channels that transport it into the filtrate. So this reabsorption secretion is accomplished by diffusion, osmosis, and in large portion by carrier-mediated transport. Whenever it comes to carrier-mediated transport, they're going to have a limit upon how much transport they can occur. That is the transport limit. That's going to determine filtration, reabsorption rates, and secretion rates. Carrier-mediated transport can occur in a facilitated manner, meaning no ATP is used. It can be active, and it can involve co-transporter or counter-transporters, meaning that both components are going the same route or both components are going in opposite routes. So we talked about renal function, so we're going to move forward from that. So this is a higher level view of where filtration occurs and where reabsorption occurs and secretion. So here we have the filtration is only going to occur in the renal corpuscle. Reabsorption is going to occur at all levels, and secretion is going to occur mostly in the proximal distal convoluted tubule and collecting ducts. Glomerular filtration, what I want you to focus here is that glomerular filtration is going to be dependent upon, pressures that are arising from the glomerulus, from the lumen of the blood vessel, basically, the pressure on the lumen of the blood vessel, and the capsular pressure, the pressure out here. The pressure in the lumen of the blood vessel is going to be higher, otherwise, we'll have movement into the blood rather than movement out of the blood. We have reverse filtration. But those are the main forces that I want you to focus whenever thinking about the glomerular filtration rates. So we have high blood pressure. There are mechanisms that allow us to regulate or buffer high blood pressure whenever it gets to the afferent arteriole. But if the pressure is higher here, you're going to have higher level of filtration occurring. This is a microscopic view of the lumen of the blood vessel, and this is the capsular space. So components are going to leave through the pores of this fenestrated capillary. They're going to pass this lamina densa, and the pedicles are going to provide these pores to which the filter is going to traverse through. So we mentioned some of the glomerular filtration rate is going to be highly regulated. One of the components that is going to play a role in this regulation, we mentioned the juxtaglomerular apparatus. It's at a place where it can sense blood pressure. It is right at the level of the afferent arteriole. So if the afferent arteriole all of a sudden just it's enlarged, the juxtaglomerular apparatus is going to sense that increase in blood pressure, and it's going to react in a way to the release of components to regulate it. So here is it's what happens whenever the glomerular filtration rate is decreased. So in this case, if the lumen of the afferent arteriole just is increasing, is decreased due to a decrease in blood pressure, you have the juxtaglomerular apparatus releasing renin. Renin activates angiotensin. Angiotensin, we mentioned that it gets activates angiotensin into angiotensin 1. Angiotensin 1 gets activated into angiotensin 2 at the lung level. And eventually this leads to a series of reactions that we're going to look closer later that elevates blood pressure and returns homeostasis. This is this is a higher, more detailed view of this process. Renin is produced, angiotensin 1 activates, and there's angiotensin 1, and that eventually gets activated into angiotensin 2. Angiotensin 2 leads to constriction of efferent arterioles. So basically, we mentioned that the afferent arteriole, the one that goes into the glomerular, and the efferent arteriole leaving the glomeruli, there is a bottleneck at the efferent arteriole, so fluid accumulates at the glomeruli, increasing the pressure. Whenever there's low the low glomerular filtration rate, the lumen of the efferent arteriole decreases even further, so there is more of a bottleneck, so the pressure increases at the level of the glomerulus. There's also the release of aldosterone. Aldosterone eventually increases the retention of sodium. We retain sodium, and we're able to retain more water as well. That leads to increased blood volume, increased blood pressure, and so forth. There's also other mechanisms that involve the central nervous system, which includes the thirst, increased water consumption, increased fluid retention. The sympathetic nervous system can also constrict the venous reservoirs. Basically, we are recruiting venous reservoirs in order to increase blood pressure, increase cardiac output, and increase vasoconstriction. Powerful constriction. So these are all components that we mentioned are going to affect the glomerular filtration rate. Let's look at specifically at the reabsorption and secretion of different components of the nephron. So the proximal convoluted tube reabsorbs 60 to 70% of the filtrate produced. This includes water and many organic components, and it secretes the secretion occurring also at this level. This is a view of what is happening at the proximal convoluted tube. So basically, we are observing sodium being reabsorbed through this, sodium and glucose being reabsorbed through this co-transporter present here. Wherever sodium goes, so that's water. Water will follow this concentration of sodium as well. We also have here CO2 being reabsorbed as well, and this is part of this buffer system, the bicarbonate buffer system present here. So there is at this level there's regulation of the pH. At the level of the root, there is this interesting physiological event called the countercurrent multiplication event. What this does is it creates these gradients that kind of feed upon themselves to promote through osmosis the movement of water. So here you have the sodium and chloride leaving the filtrate. So we are creating these gradients of sodium and chloride at the level of the ascending limb. We're going to look at specializations within the ascending limb and descending limb that make them selectively permeable to water and selectively permeable to these ions. These specializations allows the formation of this countercurrent mechanism. So here we have the descending limb, and here we have the ascending limb. I want to start at the ascending limb. At the level of the ascending limb, we have the ascending limb is impermeable to water movement, whereas the descending limb is impermeable to the movement of ions. So what's happening at the ascending limb is sodium and chloride is being moved from the filtrate and creating this gradient. You see this ever-increasing gradient of sodium chloride that as we reach the renal medulla. And what happens is wherever sodium goes, so water moves from wherever is most concentrated to wherever is least concentrated. In this case, water is most concentrated inside of the filtrate, inside of this tube, and that is because of all of the sodium chloride that's present here in the interstitial space. So water is naturally flowing outside of the descending limb, and eventually this water is reabsorbed by the vasa recta and blood vessels that are surrounding this. We also see here contributing to this concentration gradient urea. So what we observe here is that not only is sodium and chloride creating this gradient, But also urea is selectively permeable as it gets closer through the collecting ducts, as it gets closer to the medulla. So urea is leaving the collecting ducts and creating this farther adding to this gradient of concentration through urea and water selectively leaving. So eventually, the end result of it is it's highly concentrated urine. The distal convoluted tube it performs these final adjustments and selective urea solves sodium and chloride in exchange of potassium and hydrogen ions. And here we observe that view of the exchange of potassium ions in exchange for sodium and chloride. The distal convoluted tube is also going to play a role in this bicarbonate buffer system. So we secrete the hydrogen ions. So basically, we're secreting the acidic component of this buffer system, and we are gaining this precursor of bicarbonate that it's going to eventually be, this is the basic portion of it. You can receive another hydrogen readily. So along the collecting duct system, you have reabsorption and secretion, reabsorption of sodium bicarbonate urea, and secretion of hydrogen, control of hydrogen and bicarbonate ion secretion. So we're going to talk about some of the mechanisms that allow us to determine how much water is being lost. One of them is the secretion of ADH, antidiuretic hormone. What antidiuretic hormone does is, we talked about the countercurrent mechanism here that primes the reabsorption of water, basically loss of water from the filtrate, reabsorption into the blood. That is built upon this concentration gradient of sodium chloride and urea. Now, within the distal and the collecting ducts, we can also have reabsorption of water, basically water being lost from the filtrate and then being reabsorbed into the circulation. This is done through ADH. What ADH does is it promotes the presence of aquaporins within the surface of this within the surface of this collecting duct, so this tubule, and it promotes the loss of water from the filter. This is what we are observing here. Through the presence of antidiuretic hormone, we have within these portions, what happens at the distal convoluted tube, at the loop of Henle, at the nephron loop, it's business as usual. But what's added is that these aquaporins on the surface of the distal convoluted tubule and on the surface of the collecting duct that allow the further recuperation of water, let's say that way. Water leaving the filtrate, and this water eventually going from being reabsorbed into the circulation. And you can see this is just depicting how concentrated the urine gets whenever this happens. Let's go through this summary of renal function. Here we have, we observe, at these levels, we observe filtration occurring, reabsorption of nutrients and of ions as well at the proximal convoluted tubule. At the level of the loop of Henle, we have water being reabsorbed, water due to this this ion gradient that the ascending limb is creating here. This sodium chloride gradient allows water will follow this concentration gradient. Basically, water follows sodium. It's like a rule of thumb here. So water will follow, and then water will be reabsorbed into the blood. Eventually, we have mechanisms in which water can also be reabsorbed at the level of the distal convoluted tubules. and at the level of the collecting ducts. Here we have those examples. We also have um, here at the level of the distal convoluted tubules, we have sodium, potassium being secreted and sodium being reabsorbed. Um, we have that similar example happening at the level of the distal convoluted at the collecting ducts. The end result of it is this concentrated urine leaving the system. This is a summary which depicts what and where different reabsorption secretions are occurring. So this is the percentage of different components present within the filtrate. So within the filtrate at the beginning, there's no creatine, but as it moves through these several stages through the proximal convoluted tubule, the nephron loop, the distal convoluted tubule, and the collective duct, you have creatine being secreted at the proximal convoluted tubule. You also see that, let's start with water. Water gets reabsorbed at you see this big reabsorption that happens at the proximal convoluted tubule. Then the loop of Henle, there's a fair amount of reabsorption as well. Some on the distal convoluted tubule and a fair amount also at the collecting duct. That's how I want you to interpret these graphs. Urea, you have urea being it has this particular wave-like pattern that involves the release of urea at the level of these convoluted tubules. Sorry, the level of the collecting duct. This is a portion that's permeable to urea, but at the same time, at the distal at the this urea is being released near the loop of Henle, and the loop of Henle does reabsorb some of that urea. It's part of this urea cycle that allows these concentration gradients to be present. Sodium gets reabsorbed here at so it leaves the filtrate and it gets reabsorbed. It leaves the filtrate and gets reabsorbed, but some of it eventually returns to the filtrate. All right, I'm going to talk a broad level about the remaining. We talked about the physiology of the urinary system. Now we're going to talk about some of the rest of the components. So here you have your ureters, bladder, and urethra. Your ureters, peromuscular tubes. They have two two smooth muscle layers that allows them to perform peristalsis. You also have the urinary bladder, which acts as a reservoir. These are some of the components: the trigone neck, internal sphincters, and rugae. This is the male urinary bladder, and this is the female urinary bladder present here. These are the components that we talked about here. This is the trigone, which is this basically triangle between where the ureters enter and where the urethra leaves. And we have here two different sphincter. You have an internal sphincter and here an external sphincter. The external sphincter is under our conscious control, and that's the one that we hold. This is the histology of the ureter, which composed of transitional epithelium here. It has a mucosa that contains a lamina propria and two smooth muscle layers present here. And you have this connective tissue layer on the outside. The urinary bladder, once again, transitional epithelium with a lamina propria. That's part of the mucosa. You also have a submucosa, and you have these three smooth muscle layers called the detrusor muscles. The detrusor, you will have two longitudinal smooth muscle layers sandwiching a circular smooth muscle layers. This is a view of the urethra, same view. Here you have an epithelial layer and lamina propria and smooth muscle surrounding it. I want to talk about the mixed region reflex. There is a local portion of the reflex, and then there is a conscious higher level portion of the reflex. So let's start here. The urinary bladder starts getting full, and there is this tension of the urinary bladder that triggers these sensory fibers that whenever we are young and still in diapers, this is spinal reflex that eventually triggers muscles to contract the urinary bladder and release. But as we grow older, there is a central component that involves the thalamus, and this eventually leads to the conscious activation of this external sphincter that we alluded to earlier. With aging, this gets harder to control, and by now we should be familiar with all of everything that I talked about. Anyway, I appreciate your time. I hope this helps, and I will see you on Wednesday. Take care. Bye.